Hi, I'm Craig Arn, Senior Application Scientist with Sapelco, and today I'm here to talk to you about biocompatible SPME fibers. Solid phase microextraction, or SPME, was originally developed for GC applications for thermal desorptions. The concept was that you had a nonpolar stationary phase onto the fiber, and that polar compounds or compounds in a gas phase would then be trapped onto the fiber and then thermally desorbed in the GC-MS system. Today what we're doing is taking that technology and applying that towards biological applications. The biocompatible SPME fibers were derived from the traditional SPME fibers, but now what we're doing is applying it towards biological matrices. With this, we're now able to extract small drugs and polar metabolites out of biological matrices and concentrate them onto the SPME fibers using the new phase chemistries we've developed. We only need a small amount of sample to actually do a concentration onto the SPME fiber. The binder that we use really shields a lot of the larger molecular weight matrix from being extracted onto the fiber, while the particles we put onto it have a high selectivity for small drugs and metabolites. The application of biocompatible SPME fibers is really only limited to the imagination of the analyst. They can be applied to soil samples, liquid samples, living tissue samples, very viscous materials, anything the analyst can imagine. So the SPME fibers are designed to be placed into a shunt if we were using a live animal. The shunt assembly would be inserted, the fiber would then be inserted into the shunt, exposed into the biofluid, and what we would do is we'd use a syringe to draw the liquid up so it's exposed onto the fiber, remove the fiber, send that back to the laboratory, and then do solvent desorption so we can do our further analysis with that. For different types of fruits, we would take the fiber, push it through into the fleshy portion of the fruit, expose the fiber section into the flesh so we can concentrate our analytes onto the fiber, retract the fiber back into the barrel portion of the needle, and then pull that out and send it back to the laboratory for testing. For plant sampling, the process would be to take a small core sample out so we could place the fiber into it. We would then expose the SPME fiber to the porous section of the plant so we can concentrate our analytes, remove the fiber, and then send that back to the laboratory for desorption and testing. Another format for these biocompatible SPME fibers is in a tippet format. The fiber is actually adhered into the tip of a pipette tip. So now what we can do is set this up for robotics automation and for high throughput analysis. So now we can either do one sample or we can do 96 samples at the same time. So the biocompatible SPME fiber probes and the tippets really broaden the application of SPME applications in biological fluids, opening up a whole new area for concentrating analytes out of difficult matrices.